to start. Welcome, everybody. Thank you for joining the Excellence in Science Cutting Edge seminar, Use of Natural and Curated Seed Archives as Tools for Characterizing Ecological and Evolutionary Responses of Plants for Environmental Change. Before we start, we would like to invite you to keep your microphones muted during the presentation. I would like to introduce Dr. Brian Govers, Director General of the International Maize in Wheat Improvement Center, who will give a welcoming message and will introduce our special speaker of today. Dr. Govers, the floor is yours.
Thanks, Isabel, and thanks everybody for connecting. Good morning, good afternoon, good evening. As you know, this is one of the seminars as part of our Excellence in Science seminar series. We're very happy that Michael accepted to give us this talk. Want to remind you that we are in a process to design the CIMMYT strategy under the One CGIAR Research and Innovation Strategy. And the CIMMYT strategy is comprised of 6 sub-strategies: an excellence in science, excellence in operation, a resource mobilization strategy, a partnership strategy, an influence strategy, and a talent retention and management strategy. And so this seminar wants to inspire us to design a better excellence in science strategy. And therefore, we're very grateful that Michael, or also called Mike Blum, who's a professor at the Department of Ecology, Evolutionary Biology at the University of Tennessee, with research in aquatic conservation, coastal sustainability, urban socio-ecology, and public health. So maybe Michael is not what I would call a usual suspect for the audience here in CIMMYT. But he's working on socio-ecology of disasters and infectious diseases, coastal protection and restoration, and conservation and management of freshwater resources on oceanic islands. So maybe we should also connect you with our colleagues from World Fish, Mike, in that sense. But what this, of course, is really about is that Michael Blum, who also has a bachelor on integrative biology by the University of California and a PhD on zoology by Duke University, gives us an overview of his research, and it is up to us to extract concepts, methodologies, ways of thinking that can be applicable to our own world, and so extract from his presentation that what is functional and useful for us. Let's say, kick our brain a little bit for those in Mexico at 8 o'clock in the morning, and I hope sufficiently caffeinated. Those in Africa in the afternoon, take another Ethiopian coffee to really get into the presentation. And as always, those in South Asia, thanks for staying on a little bit longer after this. I hope you can join your family for evening dinner. So thanks, Mike. No more from my end. Over to you.
Thank you very much. So give me a moment and I'm going to share my screen. Let's see. Just put it into presentation mode. So hopefully you all can see this opening slide.
Yes, we do.
Okay, great. So as Isabel indicated, today I'm going to talk about natural and curated seed archives and how seed archives can be used as tools for characterizing ecological and evolutionary responses of plants to environmental change, or global change for short. As the very excellent introduction of my work has indicated, I've got a lot of different interests, including work that encompasses the use of seed archives, and in particular, natural seed archives, for understanding both plant and ecosystem resilience to global change. So that's what I'm going to focus on today, but of course, my interests, I'm sure all of your interests, span other topics, and I'm looking forward to discussing how seeds sort of play into larger questions and larger pursuits. So I'm going to start with some obvious statements, things that you're more than likely very familiar with, and then I'm going to go into more technical aspects of both the work and the concepts that I'll be discussing today. So just to start with, I'll just make a very simple statement. So as you all are very aware, undoubtedly, seeds are vital socioeconomic resources. I know that CIMMYT focuses much of your respective organizational efforts on corn, but also on other seeds and agricultural resources like wheat. And this is just to illustrate the point that seeds really undergird or support vibrant economies, both in Mexico and globally. And so in recognition of that, I thought, again, I would just note how important the work is being done at CIMMYT worldwide, both as a seed archive or gene bank, but also in operational aspects of your mission, looking ahead at how different strains of plants of economic importance can be developed further with the idea of addressing issues of global environmental change. So again, it's important to note that gene banks serve a range of different functions, and the mission isn't just solely focused on food security, but also efforts to sustain global biodiversity. So looking beyond CIMMYT, so of course CIMMYT plays an important role in this mission, but other institutions like the Svalbard Global Seed Vault also contribute to global efforts to sustain biodiversity, again, focusing on species of agriculture and economic importance, as well as from a natural perspective. And extending that one step further, there are organizations, including CIMMYT and Svalbard and the Q Millennial Seed Bank, that look at questions of how or what can be done to improve and sustain resilience to environmental change. And resilience is a very broad term that can be defined a range of different ways. Today, I'll talk about resilience in the context of being able to resist or adapt to environmental change. And again, seed banks play an important role in archiving germplasm, with the idea that not only is it important to conserve the basis of biodiversity, but also to utilize it as a tool for further development with the aim of sustaining species resilience, in particular to global change. And there have been some recent advances looking at how seed archives can be used as tools for studying how plant evolution or organismal evolution in general can contribute to resilience to environmental change. One that I'd like to note, and again, many of you are likely familiar with this, there's a project called Project Baseline. It's funded by the United States National Science Foundation, based on the premise of what's referred to as resurrection ecology, or resurrection approaches to the study of evolution and species resilience to environmental change. And the premise is very simple. The idea is to collect seeds of both natural importance and economic importance, and to do so across space and time, so that you can obtain a snapshot of what populations look like across long time horizons. So the idea here is to sample current populations and then to look 10 years ahead and sample again and again 20 years ahead, all the way up to 50 or 100 years into the future, and to continue sampling at regular intervals and at regular locations to build archives that span the chronosequences of species evolution. And the idea, again, is to use those seeds as a basis for scientific investigation, focusing on evolution through, for example, studies that involve common garden approaches, common garden experimental approaches, reciprocal transplant experiments, and then a bit more sophisticated work focusing on genomic and transcriptomic sequencing. So this effort's been going on now for roughly 10 years. It's certainly had its difficulties in terms of logistics and building these archives, but it's going well now. And again, it serves as a model for how larger organizations or institutions can think about their accessions and archives as tools or resources for doing similar work. So again, thinking about building chronosequences for the study of organismal or plant evolution. So today I'm going to focus on something similar, but instead of looking at curated seed archives, I'm going to talk about natural seed archives. And in particular, I'm going to focus on what's referred to as soil stored seed archives or soil seed banks, and talk about how natural seed banks, soil stored seed banks, can be used to study plant evolution with a focus on ecosystem resilience. So not just focusing on the organism itself, but thinking about how the plant fits into a larger ecological context. And so today I'll be focusing on coastal systems, but the premise of this work, the focus of this work is applicable more broadly. It certainly can be recontextualized according to other ecosystems that have soil stored seed banks. So this is just an illustration here pointing out what these ecosystems look like, and then the process by which we go and sample the seed banks, taking soil cores and then bringing the soil cores back to the lab, sieving seeds from the soil, and then germinating the seeds. So this is generally, again, what's referred to as a resurrection approach to the study of ecology and evolutionary biology. And the work that we're doing is very specific. So we're really addressing one particular question. And again, it's within the context of coastal ecosystem resilience to global change. And so just to, again, frame what I'll be talking about and some of the work I'll be presenting, the idea is, can rapid plant evolution improve coastal ecosystem resilience? Can it help improve the fate and potentially state or future of coastal ecosystems in response or as global change continues to unfold? So I'm going to be focusing on 4 different aspects today. So I'll tell you a little bit about persistent soil stored seed banks of the study organism that we focus on. And then I'll tell you a little bit about how we use soil stored seed banks to study rapid evolution of plants, and then how our understanding of rapid evolution is contextualized, again, addresses this question about ecosystem resilience to global change. And then I'll end with some indications of where we're going with this work, and then broadly speaking, what could be done with curated seed banks like what's being held at CIMMYT. So to begin with, let me tell you a little bit about soil stored seed banks and the plant that we focus on, which is called Schoenoplectus americanus. It's a sedge, it's a C3 sedge. It used to be called Scirpus olnei. So if you look at the scientific literature, most of the work that has been done on this is actually refers to it as Scirpus olnei. It's a model species for global change research. It's been the focus of approximately 50 years of experimentation that's been done in the United States and elsewhere, in part because it's an ecological engineer, so it builds its environment, and in particular, it contributes to soil integrity and soil accretion, which I'll focus a lot on later in my talk. It's also of interest because it's common, it's widely distributed across North America and South America. So it's not something that's particular to a specific geography. You can find it in Canada, you can find it in New York, you can find it in Florida, you can find it in Mexico. It's notable because it has both clonal or asexual reproduction, so it spreads laterally by rhizome production, but it also has annual seed production, and the seeds have a very strong or durable seed coat. So they're very small, and this is illustrated here in the pictures on the right-hand side of the slide. Each stalk or each stem carries a seed head that has approximately 24 up to almost 50 individual seeds. And the seeds effectively drop into the soil where they are produced. They don't float around on the water, they don't get redistributed, they really just get deposited in the immediate location of the mother plant. And as a consequence of that local deposition, as well as sediment deposition from tides, they get buried very quickly, and they form what's generally referred to as time stratified seed banks. So as the sediment gets deposited, this happens on a regular basis, on a daily to annual basis. And so you have these elegant, very clean seed banks that, again, represent chronosequence of change over time. And what's interesting about the seeds is, as I'll illustrate today, they persist for decades and centuries, and in some instances, thousands of years. So colleagues of mine in the geological sciences use these seeds as sources of organic carbon for carbon dating when they're studying soil horizons that date back, again, millennia. So they're highly, highly durable. They're highly persistent. So this is to illustrate the process that we take to get the soil, and then take the soil back to the lab. We section it off into regular intervals. We then use radionucleotides, particularly lead-210 and cesium-137, to date the sediment. So we have a very clear objective estimate of the chronology of sediment deposition. Then we sieve the soil to retrieve seeds, and then we try and germinate them. And this is the tricky part, which I'll talk about a bit in a moment. Once we are successful with germinating seeds, we propagate the plants for experimentation. And we do so across several generations, with the understanding that there can be complications due to long-term burial of seeds. And then once we've established experimental populations, we typically genotype plants either using conventional molecular markers like microsatellites, or we use something that looks across the genome, like single nucleotide polymorphisms, or SNP markers. But we also have advanced beyond that, where we're looking at whole genome sequencing and transcriptomics, which again, I'll mention later on in my talk. And then finally, again, once we've established our experimental populations, we do extensive experimentation, which typically involves exposure to elevated salinity, elevated carbon dioxide, elevated flooding, and then interactions between different environmental pressures. So the first thing I wanted to illustrate is the opportunities one can have to gain some understanding about population or demographic history across very long time horizons. So again, looking at chronosequences of seed burial, one can estimate the relative abundance of this species. And in fact, other species that co-occur also deposit seeds in seed banks across time. And this graph here is to illustrate how seed density, which is on the x-axis, changes across the depth of the core, which is on the y-axis. So those are centimeters on the y-axis. And then how that relates to time, which is illustrated by the blue indicators. So the 1964 mark is what we established with cesium-137, and then we have more fine resolution with lead-210. And so as you can see, we can reconstruct demographic history across more than a century of time. This work, I should indicate, is done in the Chesapeake Bay. Much of our work is done in collaboration with the Smithsonian Institute's Environmental Research Center outside of Annapolis, which again has been the site of decades of work on global environmental change. So one question that always comes up is how useful are these in terms of germination? So how far back can we germinate seeds? How old are the oldest seeds that we're able to germinate? And so this is something that we've studied extensively. And this is an illustration of some of the work that we've done across about a dozen or about 12 different germination trials, where we're looking at seed age, which here is on the y-axis, and then germination success on the x-axis, and using a statistical model based on an empirical germination trials to estimate what's the oldest seed that we could potentially germinate. And from the work that we've so far done, we can germinate seeds that are more than 100 years old. In some instances, we've had seeds that have germinated almost 200 years old. And so we have a very clear understanding of how germination rate declines over time. We understand that it's a nonlinear process. So we know that there is a steady decline in germination success, but that there are breaks, notable breaks, where you see significant declines after approximately 50 years of burial. But even though we have a significant decline at older time horizons, there's so many seeds that are available in these cores that the low germination rates is not a real limit to our ability to develop experimental populations. So again, we see seeds in hundreds, if not thousands, in soil depths that are more than 100 years old. So from this, we're able to germinate seeds, and we're able to use those again for experimental purposes. One of the things we always like to do is determine genetic relatedness or genetic variation of the plants that we've germinated from the seeds that we recover from soil cores. And this is just to illustrate some important points where we can say that the plants that are older, those that we recover from deeper soils, are genetic are genetic ancestors of plants that are from seeds that recovered in shallower soils. So again, seeds that have been deposited more recently are descendants from ancestors that created or produced seeds at older time horizons. What's also nice to note is that the plants that we have recovered from seeds, that we've germinated from buried seeds, are related to the plants that are currently growing at our sample site. So this just illustrates again that we're effectively sampling a population at a site over a long period of time, that we don't see significant replacement, or we don't see lots of gene flow from other locations that disrupt the chronosequence of genetic variation or evolution at a particular marsh, at a particular location. So again, we have a pretty clear understanding of genetic relatedness. We have an understanding of ancestry and descendancy. That's a great background to build from. We use that as a basis for then using the plants, again, revived or resurrected plants in experiments. And here, this is an illustration demonstrating that key physiological functions like salinity tolerance, which relates to the plant's ability to persist or sustain growth under stressful conditions in coastal marshes that come with sea level rise. We can say that salinity tolerance is genetically based through common garden approach. And then we can also demonstrate, as illustrated in the graph here, that salinity tolerance differs over time. So we have 4 different cohorts that we've grown in a common garden under high salinity and low salinity treatments. And we can say that the oldest populations, originating from about the turn of the 20th century, so early in the 20th century, have a very different physiology than populations that originate at more recent time horizons. And so we've done this now where we've examined physiological variation over time and how that then relates to changes in genotype frequency, determined here in the very colorful graphic on the top of this image, where we can look at, again, how the phenotype, the physiological phenotype, tracks changes in genotype, and then also tracks changes in environmental conditions. And again, because we can do so over decades to centuries, we can reconstruct long-term records of plant evolution, and we can understand what are the key environmental factors that contribute to change over time. And so in this study, we were able to illustrate that changes in salinity tolerance, or the loss or gain of function, tracks rainfall and estuarine salinity over about a 100-year period. So again, it's really important to be able to say something about organismal evolution, plant evolution, and understand how plants respond to environmental change over both short periods of time and longer periods of time. But our real interest is in understanding how plant evolution contributes to or influences ecosystem resilience to global change. And so again, we have a very good understanding of what contributes to ecosystem persistence. When you look at coastal marshes, particularly with respect to sea level rise, we understand that plants or plant performance in response to flooding or inundation is a key factor. It's a key driver in whether or not marsh can keep pace with sea level rise. So the plant in this model effectively is responding to flooding, and at low levels of flooding, it doesn't do very well because it effectively dries out. At mid levels of flooding, as illustrated in the right-hand graph here, it does very well. It contributes a very large amount of organic biomass. And then at higher levels of flooding, again, it doesn't do very well because effectively it drowns. And so there is a peak or optimal performance that varies depending on the rate of inundation. And of course, the rate of inundation relates back to sea level rise. So there's a very clear relationship. It's a quadratic relationship, so it's a little bit complicated. But again, we have a very clear understanding of how flooding influences biomass production, how biomass production then contributes to elevational change or accretion of marshes that are experiencing sea level rise. And so this has been developed, this understanding has been developed into a statistical model that allows us to simulate how responses of the sedge, the C3 sedge, can contribute to biomass production. And then we've since just recently extended the model to include or consider the possibility of evolution in response to sea level rise. And so this allows us to test the hypothesis that plant evolution in some way contributes to or alters marsh elevational change by influencing accretion rates, so the amount of biomass or soil that builds up over time. We've done this with a series of common garden experiments to parameterize or to inform the model so that this isn't just a thought experiment. This is something that is based in empirical observation. So to give you a sense of this, again, this is a little bit complicated, but this is just to give you a sense of the range of different mechanisms that we're evaluating. The null expectation or the general expectation is that plants don't exhibit any sort of evolution, don't exhibit any sort of evolutionary response, that it's all ecological. And in that respect, it's a reflection of phenotypic plasticity. The hypothesis is that there is some sort of evolutionary response to inundation. And in this respect, it's generally referred to as mean trait evolution. And then there are some more complicated scenarios that we have considered, one of which is referred to as eco-evo responses. So the evolution of trait plasticity. So it's again, a bit more complicated to consider. And then we're also very interested in understanding basic genotypic variation. So how do individual genotypes vary in response? And then how those responses change depending on environment. So these are generally referred to as genetic by environment interactions or G by E interactions. So there's a lot of things that we've considered. For today, I'm just going to focus on really a few of these things just to illustrate how trait evolution or considering trait evolution is important in understanding ecosystem resilience to global change. So we again have a null model. We have a null expectation that everything is ecological, everything is a reflection of phenotypic plasticity. We're hypothesizing that evolution, in particular mean trait evolution, is important. But there is a possibility of more complicated situations, and in particular, this idea of the evolution of trait plasticity. And so this is illustrated here in this graphic on the left-hand side. Again, the idea is that there's no evolution at all, that it's just a reflection of ecological or physiological acclimation or response to inundation. The alternative is that there is some sort of evolution occurring and that there's a range of different outcomes depending on the nature of evolution by population, by time, or some combination of time and space. So just to give you a sense of this, again, this is actually a paper that is in review right now. Hopefully it'll be accepted and published soon. This is just to illustrate the breakdown of how trait variation is attributed to ecological factors or evolution. And just to summarize it very simply, everything in blue is in some way genetically based or a reflection of evolution. So in some instances, we have traits that vary, that variation in traits is almost 50% due to evolution, either over space or time. So again, it really gets to this point that evolution can be a very important factor in how traits vary. And in marsh plants, in this particular marsh plant, what we found is that root architecture is incredibly important, both in terms of how it responds to inundation. So it evolves very quickly. It's what's generally referred to as a highly labile or adaptive trait. And here on the right-hand side, this is just to indicate the nature of the variation in root architecture, root depth distribution, how it relates back to differences in population, and that's illustrated in the pink versus green, and then how it varies over time, but in both populations. So we can say that evolution is important, and evolution is important from an ecosystem perspective because the traits that are involved figure prominently in how ecosystems respond to sea level rise. And this is again just to illustrate this point that if you forecast elevation change, and again, elevation change is important because it's whether or not a marsh will be flooded, effectively go underneath the ocean over time. If you look out to the year 2100, and the forecast that one can develop is entirely contingent on whether or not evolution is accounted for. So if you don't account for evolution, your forecasts effectively will be wrong. So just to summarize, and this is just to sort of wrap things up and to illustrate where things are going in the future. So described a little bit about century-long soil stored seed banks, how they can serve as a resource for studying plant evolution. Also illustrated the point that resurrection study designs can be a very powerful approach for studying plant evolution. And then also the idea of combining experiments with quantitative modeling, and that can be a very powerful approach for understanding how ecosystems can change depending on plant evolution. And so for the next few minutes, I just want to illustrate some potential uses of both natural and curated seed banks going forward. Again, something for you all to consider. So what we're very interested in is being able to extend our time horizons beyond about 100 or 150 years. We would like to extend that back potentially up to 1,000 years. And so it's really understanding the germination conditions to revive older seeds. And then also exploring alternative methods for revival. So not just focusing on germination, but looking at how germplasm can be used differently for both revival and propagation. We're also very interested in taking a more quantitative genetics approach. So developing pedigreed families and combining traditional quantitative genetics with genomics, again, whether it's using SNPs or whole genome sequencing, to understand not just the evolution of plants, but also to evaluate how plants interact with microbial associates, and then how those interactions, again, influence associated ecosystem functions like soil integrity, soil horizon. And then we're in thinking about this in a more complicated scenario. So I've discussed one kind of experiment, which is really just a single factor experiment focusing on flooding. We're also looking at interactions between different factors like salinity, elevated CO2, and flooding, where we're looking at more complicated scenarios of outcomes. Again, thinking about not just plasticity and trait evolution, but also these more complicated phenomena like the evolution of plasticity and genotype by environment interactions to get a sense of the range of potential outcomes from multi-factor exposure. And then finally, we are very interested in practical applications of this work. So much of what we have been focusing on is about foundational or basic scientific questions. All of this work figures into an applied context. So a lot of the work that I do on coastal ecosystems emphasizes restoration. And as we understand, plants are the basic building blocks of these ecosystems, and decisions about how ecosystems are restored revolve around what plants should be used, what seeds should be sowed. And so this figures prominently in questions about environmental stewardship, as well as socioeconomic development and human safety and well-being. So I know I've gone over a lot of work very quickly. A lot of this work is not just my own. It's important to recognize that this has been done in collaboration with colleagues at a number of different institutions. Long-time colleague Jason McLaughlin at the University of Notre Dame, colleagues at the Smithsonian as well, and also not just senior associates, but junior colleagues, postdocs, and graduate students and undergraduate students have collectively over approximately 20 years contributed to this work. And again, I'd like to recognize that much of this work has been funded by different institutions that we work at, including the University of Tennessee, the University of Notre Dame, as well as federal institutions like the US Environmental Protection Agency and the National Science Foundation. And I hope that I have left plenty of time for questions. I'm really looking forward to talking with you all about this work and then discuss related topics that you may be interested in.
Thank you very much, Mike, for this excellent presentation and very illustrative. We have one question in the chat box. For which crops, plants, all seeds in your assessment from the graphic have germinated in the range above more than 100 years, and where and at which germination rate?
That's a great question. So there are reports of agriculturally important plants that have been archived in conditions that allow for germination of seeds up to more than 100 years, in some instances up to 1,000 or more years. But those are outliers, those are unusual instances. This is a question I think is of particular importance for seed archives, whether it's CIMMYT or Svalbard or Kew. How long can you keep seeds and how long are they viable? Or do you continuously have to replace them? And so again, I think it's something that is really a focus of research and is a prominent interest across the world seed banks. But our work illustrates that under the right conditions, so cold and even anoxic wet conditions, seeds can effectively hibernate or stay viable for more than a century.
Thank you so much. Here we have another question in our chat box. It says, hi, Mike. It's really a great session. I apologize if my question is not related to this session, but may I know how many times can we plant a single seed for cultivation?
That's a great question. So that's something we deal with in our research extensively. So from what we try to do is focus on single genotypes as a starting point. And then because our plants can clonally reproduce, then we can split off and we can then section off rhizomes and then we can create effectively replicate populations of the same set of genotypes. And we've done this now where we can, from a single seed or from a single plant from that seed, we've been able to replicate it hundreds of times. And again, this is for research purposes, for the pursuit of basic research. But it's, again, something that has been considered from a practical perspective, where many of the plants that effectively function as ecosystem engineers in marshes are also cultivated for restoration. So the plant that we use is cultivated for restoration, and then some of its competitors, C4 grasses in particular, are likewise used. And so I think, again, depending on the question of interest, sometimes it's important to focus on the single genotype level as a replicate unit. But many of these plants are, again, resources for broad scale cultivation and clonal propagation.
Thank you, Mike. From Singh, it says, nice presentation. Have you worked out the genetic mechanism behind the long-term viability?
I love that question. So that's a great question. We have focused on the environmental, so far we have focused on the environmental cues that contribute to germination and viability.
Oh, sorry, Mike, something happened. We can hear you.
Sorry, so we have focused on the environmental cues so far that can contribute to long germination and long-term viability. We are currently actually looking at the genomic basis of viability. So we have just finished sequencing the genome of this sedge, and we're currently doing genome-wide association studies, so GWAS studies, looking at trait variation relative to genomic variation. And one of the traits that we're looking at is viability, seed viability.
Excellent. Thank you, Mike. It says, in continuation to this question, are there environmental conditions that influence such viability in the long term?
So we know that the seed coat is very important. We know that in seed morphology and seed physiology is important in determining viability. And so looking at other plants that occur in coastal marshes, we know, for example, that plants that produce very, very small seeds. So I showed you some small seeds, but there are plants that produce even smaller seeds. Those seeds do not persist. They effectively decompose very quickly. But there are a whole category of plants, and in particular sedges, that produce very strong or very durable seed coats that allow for seeds to remain in seed banks for hundreds or thousands of years. But beyond just the seed itself, we know that it's important for the environment to be cold and wet. So the soil it's like a refrigerator. We always say that the best place to store seeds is not in our lab, it's actually in the soil, in the marsh, because it's so cold and it's so consistent that any sort of disturbance by taking the soil back to the lab, it can affect the viability of the seeds. So we literally use the marshes as our long-term storage facilities. Where again, it's very cold, it's anoxic, so there's not much oxygen in the soils, particularly in deeper soils. So it allows for seeds to effectively remain in stasis or hibernation for a very long time.
Thank you, Mike. From Ivan Ortiz Monasterio, if seed does its breeding in one or two sites mainly, do you think that we may be modifying the root system to adapt to these conditions relatively fast and potentially affecting growth adaptation?
Yeah, so again, a great question. So we're very interested in understanding root architecture and rooting depth in particular as a trait that can influence soil integrity, soil health. And broader functions that are related to that, like carbon sequestration or carbon storage. And so we're very interested in understanding how that changes depending on the environment. So doing reciprocal transplant experiments, taking the same genotypes in one location, growing them in another location, and vice versa, doing the same for other populations, and seeing how outcomes relating to soil really are contingent on that genotype by environment interaction. So we found some very consistent responses across populations that suggests that mean trait evolution is something that can occur very broadly from a geographic perspective. But we also know that there's a lot of dependencies or contingencies in response. And so really understanding this genotype by environment interaction is really where we're very much focused going into the future.
Thank you very much, Mike. We have another question here. Are historical populations likely to exhibit enhanced fitness to future environments? Do future environments have to resemble past ones for this to be a viable restoration strategy?
Another great question. So it's great that people are responding so well to this topic. So what's interesting about the trends in environmental change, so the future in some instances is, even in the near-term future, in some instances is going to be like the near-term or less recent or more distant past. And one very good example of this is estuarine salinity, or the amount of salinity stress that might act on a plant. So everybody's general conception broadly is that sea level is rising, and with sea level rise, the expectation is that there will be a linear increase or potentially exponential nonlinear increase in flooding and salinity exposure. But what we found is that salinity in estuaries across coasts is highly dynamic, and it's a much more complicated situation that is contingent both on sea level rise and precipitation in coastal areas. And so this balance between precipitation and sea level rise can make the future look like the past. So you see nonlinearity, you see rises and falling trends in salinity conditions because you have these complicated interactions. So this is something we found across the 20th century in the Chesapeake, where we found long stretches of time late in the 20th century that were like the early 19th century. And so again, it's important to think about how these environmental factors can potentially interact to understand what the future is going to look like.
Thank you so much. Okay, here we have a comment from Zinc. It says, it did not happen for CIMMYT. Using selection environments in Oregon for initial selection and testing worldwide, in fact, broadened the wide adaptation. So
yeah, that's interesting. Mm-hmm. So that's definitely something that can happen. And that's something we're very interested in understanding, how different traits can respond to different kinds of selection, and how different factors that act as selective agents can create something called a geographic mosaic of selection. So the best way to think about this is that there are global factors that are changing the same way across the world. Elevated carbon dioxide, for example, there's slight variations depending on where you are, if you're in North America versus sub-Saharan Africa, but the general trends are consistent globally. But then you have local factors like precipitation or nitrogen availability that change at very local scale. And so there's real strong interest in understanding how plants can evolve to global factors like CO2, and then how that then interacts with responses to local factors like nitrogen availability. And so that's where looking at these multi-factor experiments can start to differentiate between how a plant can adapt very broadly, but then how these local adaptations then can mitigate or in some way mediate those global adaptations.
Thank you, Michael. Thank you very much for this interesting conversation. Would you have an audience? I would like to invite my colleagues that are in this session connected. Whether anyone else would like to make another question or comment, you can raise your hand or you can write it in the chat box. Okay. Well, I just want, okay, here we one more question. It says, if long and deep-rooted wheat variety may be developed, it will be applicable for the areas where water is shortage. Is it possible to develop such type of variety?
So my answer to that is yes. So this is one of the really interesting things that we're finding is that much of the work so far on plant evolution or response to selection has been on in the above ground traits like stem height or stem density. And it's been much more difficult to look at below ground responses in root architecture, root biomass. And so it's something that, again, really stands out as an opportunity for further investigation and further application is focusing on below ground traits. So we found in our plants, in our sedges, that rooting depth was something that responded to selection much more quickly than almost any other trait. And so it has a lot of what's generally referred to as adaptive potential or adaptability. And I would say it's not particular to the sedge that we're looking at. Rooting depth and root architecture is something that we understand is a very labile trait and can respond to selection in other plants that we've looked at. And so I would say that one of the lessons from our work is that more work on rooting architecture can lead to amazing practical outcomes. So whether you're interested in marsh elevational change and whether marshes are going to be flooded by sea level rise, or you're interested in soil health or water shortages, again, I think it's something that presents an amazing opportunity to focus on.
Thank you, Michael. Well, it looks like a very interesting conversation and very educative information as well. Here we have another one. Are there curated archives for non-crop plants?
Yeah, so there are a few. So the Kew Royal Garden in the United Kingdom is one example. So one of their mission areas is to develop curated archives of all native plants across the United Kingdom. And I think they've just achieved that. But then there are other smaller scale archives. So for example, Project Baseline that focuses principally on non-crop plants. But I think, again, thinking about the plants as tools can help facilitate interest in developing seed archives and thinking about how plants can serve as ecosystem engineers, and in particular for soil health and soil integrity.
Thank you, Michael. Thank you very much, Michael. Would you like to give a final comment for this interesting presentation before we close the session?
Yeah, so again, I'd like to thank everybody for participating in the discussion today. And I'd also like to thank Dr. Jay Gulich for recommending me as a speaker. It's been a fantastic opportunity to talk with you this morning. And I'll say that, again, much of my interests from a science perspective don't necessarily focus on seeds, but I find seeds amazingly fascinating. They're just really amazing. And I think that there are some tremendous advances that can be made both in fundamental science, as illustrated in some of the work that I talked about today, but then also how particularly curated seed archives can be used for different kinds of applications. So certainly food security is one very, very important mission to focus on, but there are associated areas or pursuits that can be addressed that complement that primary focus. One, again, is soil integrity. We're factoring in issues like drought or salinization of soil. So I think, again, broadening out the focus on global food security and thinking about environmental factors and how that relates to soil and how plants can figure prominently as a tool for not necessarily manipulating ecosystems, but engineering conditions so that they become more favorable. We've certainly seen that in marshes. Marshes are important globally because they figure prominently in global carbon cycling. But again, it's not particular to marshes. It's something that can be addressed in other ecosystems of interest. And it doesn't have to be natural ecosystems. It can certainly be managed or agricultural ecosystems.
Thank you very much. Thank you so much, Michael. On behalf of the International Maize and Wheat Improvement Center, I would like to give our gratefulness for this very interesting seminar, Use of Natural and Curated Seed Archives as Tools for Characterizing Ecological and Evolutionary Responses of Plants to Environmental Change by Michael Blum, professor at the Department of Ecology and Evolutionary Biology at the University of Tennessee. Thank you very much, Mike, for your valuable time. And thank you very much for the audience. Have a nice day and nice evening or nice morning.
Thank you all.